We often hear that our genes determine who we are. They are like the blueprint of our bodies, the fundamental code that shapes our physical and even some aspects of our mental traits. Our genes dictate our hair color, our height, and even our risk for certain diseases. They are the reason why some of us have blue eyes while others have brown, why some are tall and others short, and why some may be more prone to conditions like diabetes or heart disease. But what if I told you that there's another layer of information on top of our genes? This additional layer is not about changing the genetic code itself, but about how that code is read and interpreted by our cells. This layer is called the epigenome. The epigenome consists of chemical compounds that modify or mark the genome in a way that tells it what to do, where to do it, and when to do it. Think of your genes as a giant instruction manual. This manual contains all the information needed to build and maintain your body. The epigenome acts like a series of sticky notes all over that manual. These notes provide additional instructions, highlighting certain sections while ignoring others, ensuring that the right information is used at the right time. These notes don't change the words in the manual, our DNA sequence, but they tell our cells which pages to read and which to ignore. This selective reading is crucial for the proper functioning of our cells and ultimately our bodies. These sticky notes are chemical tags that attach to our DNA. They can be added or removed in response to various signals, acting as a dynamic system of regulation. They can switch genes on or off without altering the underlying genetic code. This ability to turn genes on or off is essential for processes like cell differentiation, where stem cells become specialized cells like muscle or nerve cells. This process is called epigenetics. Epigenetics is the study of these chemical modifications and how they influence gene activity. Epigenetics is like having a dimmer switch for our genes, fine-tuning how they are expressed. Just as a dimmer switch can adjust the brightness of a light, epigenetic changes can adjust the activity level of a gene. Epigenetic changes can happen throughout our lives due to various factors. These factors can include aging, lifestyle choices, and environmental exposures. Our diet, environment, and even our experiences can leave epigenetic marks on our genes. For example, what we eat can influence the addition or removal of these chemical tags, potentially affecting our health and well-being. This means that we are not simply slaves to our genes. We have a surprising degree of control over how our genes are expressed. By making conscious choices about our lifestyle and environment, we can influence our epigenome and, in turn, our health and longevity. One area where epigenetics plays a crucial role is in sex determination. This fascinating field of study delves into how our genes are expressed and regulated, influencing whether we develop as male or female. While our sex is determined by our chromosomes, X for females, XY for males, epigenetics helps translate this genetic information into physical characteristics. This process is not as straightforward as it might seem. The interplay between our genetic code and epigenetic markers is complex and finely tuned. You see, both males and females carry the genes for both male and female characteristics. These genes are present in all of us, but how they are expressed depends on epigenetic regulation. This means that the same genetic blueprint can lead to different outcomes based on which genes are turned on or off. Epigenetic marks, such as DNA methylation and histone modification, help silence the genes that are not needed for our biological sex. These marks act like switches, turning off certain genes while allowing others to be expressed. This selective gene expression is crucial for the development of sex-specific traits. For example, in females, epigenetic marks silence genes responsible for developing male genitalia. This ensures that the female reproductive system develops correctly with all the necessary structures and functions, while in males, similar marks silence genes for developing female genitalia. This allows the male reproductive system to form properly with the appropriate organs and characteristics. These epigenetic marks ensure that the right genes are turned on or off at the right time during development. This timing is critical as the development of sexual characteristics follows a precise schedule. Any disruption in this process can lead to developmental abnormalities. This intricate dance of gene regulation is essential for the proper development of sexual characteristics. The coordination between genetic and epigenetic factors ensures that our bodies develop according to our genetic blueprint, but with the necessary modifications for our biological sex. Without it, the boundaries between male and female could become blurred. This could result in intersex conditions where individuals have characteristics of both sexes. Understanding epigenetics helps us appreciate the complexity of sex determination and the delicate balance required for normal development. But epigenetics doesn't just influence the development of our physical sex characteristics, it can also influence our behavior. The same mechanisms that regulate gene expression in our bodies also affect our brains, potentially shaping our thoughts, emotions, and actions. We see this in the animal kingdom, where epigenetic changes can influence mating behaviors and even parental care. For instance, certain epigenetic modifications can make animals more nurturing or aggressive, depending on the environmental cues they receive. This suggests that epigenetics might play a role in shaping human behavior as well. Our experiences and environment can lead to epigenetic changes that affect how we think and behave. This opens up new avenues for understanding the biological basis of behavior and the potential for epigenetic therapies in treating behavioral disorders. The relationship between males and females is complex. In evolutionary biology, this complexity is often understood through the lens of sexual conflict. This means that the evolutionary interests of males and females are not always aligned. Males and females have different strategies for passing on their genes. Males can maximize their reproductive success by mating with as many females as possible. Females, on the other hand, invest heavily in each offspring and therefore benefit from being choosy about their mates. This difference in reproductive strategies can lead to conflict. For example, males might evolve traits that help them compete for mates, even if these traits are detrimental to females. Conversely, females might evolve strategies to resist male control over mating, even if these strategies are costly to males. This evolutionary tug-of-war between the sexes can have a significant impact on the traits that evolve in both males and females, and as we shall see, epigenetics might play a crucial role in this ongoing battle. Dr. William Rice, an evolutionary biologist, has proposed a controversial hypothesis suggesting that epigenetics might play a role in homosexuality and transgender identity. This hypothesis has sparked significant interest and debate within the scientific community as it challenges traditional views on the origins of sexual orientation and gender identity. He argues that some epigenetic marks, instead of being erased between generations, can be passed down from parent to child. These marks, which are chemical modifications to DNA that do not change the DNA sequence itself, can influence how genes are expressed. This means that the experiences and environments of parents could potentially impact the genetic expression of their children in ways that were previously unrecognized. According to Dr. Rice, these inherited epigenetic marks could influence a child's sexual development. This idea is based on the understanding that epigenetic marks can affect the sensitivity of cells to hormones, which play a crucial role in sexual development and differentiation. For instance, the timing and levels of hormone exposure during critical periods of development can have lasting effects on an individual's sexual orientation and gender identity. For example, an epigenetic mark that promotes testosterone sensitivity, if passed down from a mother to a daughter, might increase the likelihood of same-sex attraction in the daughter. This is because heightened sensitivity to testosterone could influence the development of brain regions associated with sexual preference. The intricate balance of hormone levels and receptor sensitivity is a key factor in shaping sexual orientation. Similarly, an epigenetic mark that reduces testosterone sensitivity, if passed down from a father to a son, might increase the likelihood of transgender identity in the son. Reduced sensitivity to testosterone could affect the development of secondary sexual characteristics and brain structures that are typically influenced by this hormone, potentially leading to a gender identity that differs from the sex assigned at birth. It's important to note that Dr. Rice's hypothesis is just that, a hypothesis. While it is grounded in scientific principles and supported by some preliminary evidence, it remains to be thoroughly tested and validated through rigorous research. The scientific method requires that hypotheses be subjected to empirical scrutiny and replication before they can be widely accepted. More research is needed to determine if epigenetic inheritance plays a role in human sexual orientation or gender identity. This research would involve studying large populations over multiple generations, as well as conducting detailed molecular analyses to identify specific epigenetic marks and their effects. Such studies are complex and resource-intensive, but they are essential for advancing our understanding of these phenomena. However, the hypothesis highlights the potential for epigenetics to influence complex human traits. Epigenetics is a rapidly growing field that has already provided insights into a variety of biological processes and diseases. By exploring the role of epigenetics in sexual orientation and gender identity, scientists may uncover new mechanisms that contribute to the diversity of human experiences and behaviors. It is crucial to approach this topic with sensitivity and respect. Discussions about sexual orientation and gender identity can be deeply personal and emotionally charged. It is important to recognize the lived experiences of individuals and to avoid stigmatizing or pathologizing these aspects of human identity. Scientific research should be conducted and communicated in a way that promotes understanding and acceptance. Sexual orientation and gender identity are complex aspects of human identity, and we are only beginning to understand the interplay of genes, environment, and individual experience in shaping them. As research progresses, it is likely that we will discover even more intricate interactions between genetic, epigenetic, and environmental factors. This knowledge has the potential to enrich our understanding of human diversity and to inform more inclusive and supportive approaches to health and well-being.